friends, welcome to today's class. Today we are going to study about DNA replication under the subject Biochemistry BP203T. In the previous lectures we have studied about the nucleotides, nucleosides and about the structure and functions of DNA and RNA. Now let us go through the central dogma of molecular biology's steps. Like the first step is DNA replication. So today we are going to study about DNA replication. What is DNA replication? It happens in DNA only, as the name suggests. And about the DNA, let us see some important points of DNA. Like what is DNA? DNA is a macromolecule that carries the genetic information from generation to generation. It is the reserve bank of genetic information. That means it reserves, it has the memory of our previous generations which comes up to us and then will transfer into our next generation. And it is composed of two polynucleotide chains, that coils, that means that helix in the structure of helix around each other to form a double helical structure, and it carries the genetic instructions for the development, for the functioning, growth and reproduction of all known organisms in this universe. Then the thing is RNA. What is RNA? RNA is again ribonucleic acid. It is the polymeric molecule essential for biological roles in coding and decoding, whereas regulation and expression of genes also happens by RNAs. Next, RNAs are generally single stranded. They contain ribose sugar, whereas DNA contains deoxyribose sugar. RNA, there are three types of RNA we know, that is, first is mRNA, also known as messenger RNA. What is the function of mRNA? mRNA serves as a temporary copies of information which founds in DNA. Next is rRNA. rRNA is also called as ribosomal RNA. It is the structural component of protein making structures known as ribosomes. We have heard about ribosomes many times, and here with rRNA we are going to study about some of them. Then tRNA. tRNA is called as transfer RNA. It carries the amino acids to the ribosomes which needs to be assembled. So these are the working of mRNA, rRNA and tRNA. And in the previous slide we have discussed about central dogma of molecular biology and also called as central dogma of life. So let us revise it once again. Central dogma of molecular life or central dogma of molecular biology, that means there are three steps followed in here. That means the DNA will replicate itself and the process will called as replication. In the second step, DNA will form RNA. That means DNA forms RNAs, the process will called as transcription, and the last process, that is RNA, will form protein, and that is called as, the process will be called as translation. Then the biological information that flows from DNA to RNA and then to protein is the central dogma of life. DNA is duplicated, that means it is replicated, maintained and passed down accurately to the daughter cells. That means whatever DNA has, whatever coding it has, whatever nucleotide sequence it has, it will replicate itself and will transfer the same thing to its daughter cells. Then for fulfilling these functions, it should follow three type of R's. What are those? First, it should do replication, then it should do recombination and it should do repair. As we all know, DNA, when it is replicating itself, it should, if there is any kind of mistakes happen, then it should repair itself simultaneously, because incorporation of a single strand, incorporation of a single nucleotide will become mutation, will become drastic problem in the daughter cell, and then it will replicate likewise only. So, there should be repair process when replication is happening. So, now let us start with our replication process. In replication process, the parent DNA, which is helical, has two strands. Each of the strands of DNA will form daughter cell. That means, if there are two strands of DNA present in a parent DNA, then those two strands will make 4 daughter DNA. That means, in total it will make 2 daughter DNA. That means, if we can see, there are two strands, in this strand we have seen that there is one strand here and the second strand here. That means, these two strands will make, this one strand will make one daughter cell and the second strand will make another daughter cell. Okay. So, here that means only one single strand of DNA will require to make a complete daughter cell, daughter DNA. So, with one parent DNA we can make 2 daughter DNAs. Then, how this replication process happens? Replication is a process in which DNA copies itself and produces identical daughter molecules of DNA. It should be with high fidelity. That means, it should be with proofreading. That means, if there is any repair mechanism needs, DNA will repair itself at the same time, so that the mistake cannot be repeated several times, the mistakes cannot be transferred to the daughter cells and it will be transferred so on. So, the correction should be made at the same time if the DNA will think there is a mistake happening. So, now the process of replication of prokaryotes. As we discussed in this slide, that only half of the DNA, parent DNA, will make one full daughter DNA and the other half of the DNA will make the second daughter DNA. That means, at a time we are using just half of the parent DNA. That is why it is called as, the replication process will be called as semi-conservative. That means, we are conserving one of the parent strand of DNA and the next strand we are using for making the daughter cells. So, the parent DNA has two strands which are complementary to each other and both the strands are undergo replication to produce two daughter molecules. What do you mean by complementary? Complementary means that they are having these are the two strands of DNA. Now, if the first and second strand, if first strand having A, that means the second strand will have T, the first strand will have G, then the second strand will have C and so on, and likewise that. Okay. So, they are complementary to each other, they are not identical to each other, they are complementary. This function is called as complementing each other. So, both the DNA strands are complementary to each other and finally they produce two daughter molecules. Each one of the newly synthesized daughter molecule will again do the same thing and then they will form next generation, next daughter molecules. The next daughter molecule will do the same thing and they will form their next generation and likewise they will replicate themselves. And it was at the first experiment was done by Messelson and Stahl. Then only we got to know that the DNA replication is a semi-conservative process. Now, from where this DNA replication happens, how it will replicate itself, now we are going to study about the process. DNA replication will not start from anywhere. There are certain spaces, certain positions from where DNA replication will start and those are called as ori. ori is also called as origin of replication. So, there are certain places in full DNA that are called as ori or origin of replication, from where the replication will starts, initiates. Okay. So, from here, like we have one ori here, we are one here and we are one here. So, there are certain ori means origin of replications present in the DNA molecule, and then from there only the replication will starts. That means, if you are seeing the red lines inside, those red lines are the newly forming daughter cells. The blue lines, these blue lines are your parent DNA, and the red lines inside those parent DNA is your newly formed daughter DNA, which are in the process of forming. That means, the process is initiating just. Okay. In the next step, or in the next slide, we are seeing that there is a protein. How this, now let us discuss about how this replication initiates. So, there is a specific protein called as DNA A protein, which go and binds to the space, binds to the ori, and from there, there is a initiation of DNA replication. So, you can see this is an ori, this particular circle is the ori, that means this is from where the replication will start. Here the DNA A protein is attached in somewhat places, and from here the synthesis of newly daughter DNA forms daughter DNA from this place only. So, what do we understand till now? That the DNA has two strands, the parent DNA has two strands. Each strand of that parent DNA will form a daughter DNA. That means, one parent DNA will form two daughter DNAs, and randomly from nowhere DNA replication will not start. There should be the origin of replication for every DNA molecule, for every parent molecule to make their copies, and in the origin of replication, a protein known as DNA A will act upon, and from there the initiation will start. Now, let us understand. We know that DNA is a helical structure. So, for that, for doing the replication, we need to separate the parent DNAs. Now, if there is a helical structure of DNAs happening is there, so how we are going to make the daughter cells in that? So, for that the DNA needs to unlock themselves, DNA needs to unhelix themselves, and then only there will be the formation of new DNA molecule. So, when DNA molecule, likewise here we can see DNA, the parent DNA already has separated. So, there are two parallel lines here indicating the DNA molecule. In this two parallel lines, there is a ori, that is origin of replication, and in the third structure we are seeing that there is already been started initiation of daughter DNA synthesis. So, now we are seeing here leading strand and lagging strand. Now, let us understand what is leading strand and what is lagging strand, and here are some numbers are written like 5 dash, 3 dash. What are those? First of all, we need to know that DNA replication always happens from 5 dash to 3 dash, from 5 dash to 3 dash. That means these are the directions, from which they needs to be synthesized, newly synthesized DNA will be happening in this direction. What is leading strand? Leading strand means when there is the synthesis of daughter DNA in continuous form. That means the DNA will form in the continuous form like this. The new DNA will synthesize like this way from 5 dash to 3 dash. And then about the lagging strand means there will also be DNA synthesis, daughter DNA synthesis, but because as I told you that synthesis will only happens from 5 dash to 3 dash, Now here is a problem. In the lagging strand there is a problem, because now both the strands, both the parent strands are complementary to each other. So one parent strand, if it is from 3' to 5', the other parent strand will be from 5' to 3'. And as I have told you that the synthesis of newly DNA strand will happen from 5' to 3' only. So it is okay with the leading strand, because we can see there is newly DNA synthesized will be from the direction 5' to 3'. But in the lagging strand, because it is complementary to the parent DNA, the first strand, so here it is very difficult for making the 5' to 3' strand continuously. So there will be small fragments like this. One small fragment will form, then again one small fragment will form, then again one small fragment will form. So these small fragments will form on the lagging strand, because the direction 5' to 3' is not happening here continuously. That is why we need to do this on the lagging strand, and these small fragments later on at the last will be joined together and a new daughter DNA will be formed. In the leading strand it is happening continuously, but on the lagging strand it is happening in discontinuous form. Okay. These small discontinuous fragments, newly synthesized DNA, are called as Okazaki fragments. These are called as Okazaki fragments. Then let us again discuss the whole thing. I have mentioned important points of DNA replications in this structure. Now let us discuss what is replication fork. As I have told you that DNA needs to be separated through the helical structure. It needs to be two parallel lines of DNA to do the replication. Now it is very difficult to separate the DNA when it is helically formed, when it was helically formed. Now, so how to do it? Then if you have seen certain threads, certain strings, or zipper of your jacket, zipper of your shirts, now what you have done, what you have seen, that when you are opening the zipper, at one point the zipper is opened, that means there are like this fashion, this means fashion, and the zipper, from where the zipper is not opened, it is a single ended line. So there is this type of structure happens like that. So when you have opened, this is the part where you have already opened the jacket, and this is the part which you are going to open the zipper. Now, so this is called as fork structure for DNA replication. This is the same structure. That means we have unhelixed the DNA structure, and now we are starting with the DNA replication. So certain enzymes are there which needs to open the helical structure and then to stabilize the structure. It is very important to stabilize the structure. So as you have seen, 3' to 5', and the second strand 5' to 3', that means they are complementary to each other, that is why they are like that. Okay. Now this is your leading strand, this is your leading strand, let me, and this is your lagging strand. Now what is happening here? We have started from this place, that DNA synthesis, the newly DNA daughter DNA will synthesize from here only. So this will be, I told you, 5', because it will be complementary to the previous one, and a continuous synthesis of DNA will happen here. That means as you are opening the zipper, the synthesis will happen a long way. But in the second strand, what is happening? Like if you have opened this much zipper from your jacket, so only this part has synthesized. You have again opened the zipper, again this part has synthesized. Again opening it, this part has synthesized, and so on. Now, as you have seen, if there is something in helical structure and you try to open it, and if then you lose it, it will again forms the helical structure, which we have seen very commonly in the thread that we are using at home, that if you open the helical structure of the thread, as soon as you release the tension, it will again become a helical structure. So, there is very important to stabilize at this point. Once you have opened the structure, it should be stabilized. So, now which proteins are used here? First of all, to open the DNA molecule into a fork, we need here helicase. DNA helicase is required to open the helical structure into a fork. Then the second, which needs to be stabilized the opened DNA molecule, that means we have opened the helix like that and we do not want to re-helix it once again. So, we will use here some SSB protein. SSB proteins are called as single stranded binding proteins. Here they are used to stabilize the open structure of your DNA, parent DNA actually. And the newly synthesized DNA, it is synthesizing with the help of enzyme DNA polymerase 3. That means when there is a synthesis of newly synthesized, that means the replication process is incomplete without DNA polymerase 3. Now, these are the, as I have told you, these are the Okazaki fragments, the small fragments, and later on they will joint together. And here to stabilize the zipper part, the completely zipped part is stabilized by another enzyme, is your topoisomerase 2, DNA topoisomerase 2. Let us discuss these strands. Let us discuss some points which we have discussed in the previous slides. That what are Okazaki fragments? Okazaki fragments are those small strands on the lagging DNA strand, and later on they are joined by DNA polymerase 1 and DNA ligase. DNA ligase is the enzyme which helps to stick the Okazaki fragments together and forms a big normal daughter DNA. Then the second is initiation. In prokaryotes, the initiation of DNA synthesis replication occurs at a site called as origin of replication, single site in case of prokaryotes, whereas it will be multiple in the eukaryotes. We are discussing replication in prokaryotes right now. So, the next point is DNA A protein is used for the starting of your origin of replication. Then the two complementary strands of DNA separates at the site of replication to form a bubble in prokaryotes and multiple in the eukaryotes. RNA primer is required for the synthesis of new DNA. That means, let me tell you, when we are starting with here, the very first the strands which we are synthesizing is happening with the help of RNA primer. That means RNA primer is there to start the initiation process, and the length will be increased with the help of our DNA polymerase 3. Okay. So, DNA A protein, that is around 20 to 25 monomers, binds at the site of origin for replication. This causes double stranded DNA to separate. The two complementary strands of DNA separate at the site of replication to form a bubble, and RNA primer, that is 5 to 50 nucleotide, is required to form to start the initiation process. We need SSB protein for the stabilization, and a constant synthesis will happen on. Then DNA synthesis is a semi-continuous and bidirectional. As I have told you, it is semi-discontinuous, means for the leading strand it is happening in continuous fashion, but for the lagging strand it is happening in discontinuous fashion. Small fragments are forming, Okazaki fragments are forming, and later on they will be joined together. And why it is called as it is bidirectional? Because the two DNA strands are complementary to each other. The parent strand first is 5' to 3', definitely the second will be of 3' to 5'. So, when there will be synthesis happen in one, the synthesis will happen in this direction, whereas the synthesis will happen in this direction. That is why it is bidirectional. DNA helicase is the enzyme which is used as zip opener, and it is a ATP dependent enzyme. It needs energy to do that function. SSB proteins, we have discussed that DNA helix destabilizing protein. It will not help the DNA to become the single stranded, to become stabilized in that form, and DNA polymerase 3 catalyzes the synthesis of new DNA strand. It happens in 5' to 3'. Then it also does proofreading. As I have already told you, proofreading is a very important process in the DNA synthesis, DNA replication, because if there will be any incorporation of wrong DNA, wrong nucleotide, then it will then create so many problems. Mutations can be there. DNA polymerase 1 and DNA polymerase 1 and DNA ligase, these Okazaki fragments are lastly are joined by these enzymes. DNA polymerase, it helps in proofreading. DNA polymerase edits its mistakes and removes the wrongly placed nucleotide bases. Then at last there will be replacement of that RNA primer with the DNA molecule, because we have initiated the synthesis with the RNA primer. Though that RNA primer needs to be edited out at the last. And the enzyme DNA ligase, that forms a photodiester linkage between DNA synthesis by DNA polymerase 3 and small fragment of DNA produced by DNA polymerase 1. That is, this process is called as nick and seal method. DNA polymerase 2, it helps in the DNA repair process when there is a need to remove the wrongly attached nucleotide base. And then DNA topoisomerase helps in the overcome the problem of supercoiling. That means, if we have opened, like we have made this Y-shaped structure a fork, then the process, then the part which is not open till now will come like that, and again there will be a helix. So, to do that, to solve this problem, this part, this unopened part will be opened at one place and maximum supercoiling will be removed, and again it will be done like that. So that to overcome the problem of supercoiling, DNA topoisomerase is used, and DNA gyrase is an enzyme which cuts and reseal to overcome the problem of DNA supercoils. This is the replication eukaryotes. It is almost the same, but as we have know that eukaryote is a typical organism than prokaryote. So, it has several more enzymes to which, that is DNA polymerase alpha. There is a synthesis of RNA primer, both leading and lagging strand. DNA polymerase beta, it helps in DNA function to compatible with. It has DNA polymerase gamma, it helps in the replication of mitochondrial DNA. DNA polymerase delta, it is responsible for replication of leading strands. DNA polymerase alpha, beta, gamma, delta and epsilon, that is, it helps in 3 steps, that is initiation, elongation and termination. So, it is a little bit different than eukaryote, otherwise rest of the processes are same as in DNA replication for prokaryotes. In the leading strand, here the DNA polymerase lambda and a sliding clamp called as proliferating cell nuclear antigen is used, and pCNA forms a ring around DNA to which DNA polymerase lambda binds. Formation of the ring also requires another factor, namely replication factor C, that is called as RFC. This is in the lagging strand. It is the same as in prokaryote, but certain things are different, like there is an incorporation of an replication protein A. In that we had DNA A for starting the replication process. Here we have replication protein A. The enzyme primase is used, same as initiates the synthesis of Okazaki fragments. DNA polymerase A elongates the primer by addition of 20-30 deoxyribonucleotides, and finally we will have our DNA strand for eukaryotes. So, in the next step, the binding of replication factor C is to elongate the primer, the DNA polymerase delta to bind the sliding clamp and elongates the Okazaki fragments. The RNA primer removal will be done by RNA H and flap endonuclease. Look, I have told you that there are some different enzymes than DNA replication in prokaryotes. Ligation is done by DNA ligase only. This is the full steps which are written in the language there. So, this is the full step, we can see through it that how this is happening here, what are the inhibitors in the replication process. So, there are certain antibiotics which helps in the inhibition of replication process, that are normal ciprofloxacin, novobiacin, nalidixic acid. They blocks the replication of DNA and multiplication of the cells. They do not affect human enzymes, and certain compounds which inhibit human topoisomerase, they are also used as anti-cancer agents like adrimycin, etoposide, doxorubicin, and other anti-cancerous agents are also there. These are the references which I have used. Thank you so much.